now we continue with the short talks. Our first speaker, Tom Hindmarsh, will be talking about dissociating task acquisition from expression reveals latent knowledge.
Thank you. I'm excited to kick off the contributing talks for this session and to be here at Cosyne. My name is Tom Hindmarsh Sten. I'm a graduate fellow at the Rockefeller University in Vanessa Rutta's laboratory. But today I want to talk about some work I did as an undergraduate with Kuchibhotla's lab at Johns Hopkins. And this work really stems from the observation that assessment of learning and aptitude in animals and humans requires a subject to report their underlying knowledge at a given moment in time. The performance of a student who's been studying for a test, for example, is assessed at a discrete moment, and the student's performance at that moment is taken as a kind of proxy for how well they've retained information. And a big driving factor for such performance is, of course, learning, both in the form of sensory discrimination abilities and the formation of accurate sensory motor associations. But contextual factors that emerge from the kind of behavioral setting of the task, such as the sensory environment or the availability of rewards, also strongly impinge on behavior. And these can be adaptive, as in the case of foraging strategies, or maladaptive, as in the case of anxiety. And while interesting in their own merit, they're fundamentally unrelated to the subject's knowledge of the core task demands and may kind of confound any interpretation of behavioral performance. So what I want to share with you today is some of the work we've been doing trying to dissociate the acquisition of underlying learning from the kind of contextual factors that constantly impinge on behavioral decision-making. And what I hope to show you is that the acquisition of underlying knowledge actually occurs far faster than behavioral performance often suggests. And instead, the performance during learning is driven more by the testing context. And second, via theoretical modeling and some neural recordings, we identified two parallel learning processes, one related to the acquisition of underlying knowledge and another related to the expression of that knowledge regardless of the testing context. So at first we trained head-fixed, water-deprived mice on an auditory go-no-go task. Mice are trained to lick to a target tone for a small water reward and to withhold from licking after a foil tone to avoid a short timeout. And mice learned this task very well over a number of days, about 2 weeks in total. And at expert performance levels, they lick consistently to the target tone and not to the foil tone. So a typical animal would look something like this. The mouse licks to the target tone, licks to the target tone, but doesn't lick to the foil tone. But to understand how context impacts the kind of acquisition versus expression of knowledge, we focused on the earlier stages of learning. And throughout learning, we interleaved something we call non-reinforced probe trials, where we remove the lick tube and the animal's access to reinforcement. In a typical training session consisting of about 300 reinforced trials, in the middle we insert 20 to 40 trials where we remove the lick tube. And at first we focused on a day early in learning where animals were tone responsive, but they were not discriminating. So a typical animal at this stage of learning would look something like this in the presence of reinforcement. The animal licks to the target tone, but also licks to the foil tone. And notice that he's not licking indiscriminately between tones, but selectively when tones come on. Again, he licks to the target tone, but also to the foil tone. So this animal essentially shows zero discriminatory abilities and is a chance level. When we remove the lick tube and the animal's access to reinforcement, we see a different story. This animal licks to the target tone, licks to the target tone, but he doesn't flinch to the foil tone. He licks to the target tone, doesn't lick to the foil tone, licks to the target tone. So despite this animal's poor performance in the presence of reinforcement, he shows us that he has understood the full kind of task contingencies many days before his performance in the presence of reinforcement will reach expert levels. And if we look across animals at this day in learning, we see that animals consistently discriminate better between the two tones in the absence of reinforcement than in its presence. So here the hit rate stays constant, whereas the false alarm rate, the indiscriminate licking to the foil tone, essentially plummets in the absence of reinforcement. So while we see a slow and gradual learning trajectory in the presence of reinforcement, we see a much steeper learning trajectory if we look at the animal's behavior on these probe trials over the course of learning. And we think that this kind of serendipitously allows us to dissociate the acquisition of underlying task, understanding the underlying task contingencies from the expression of that knowledge. And then we kind of asked to what extent this simple behavioral manipulation, the withholding of reinforcement, could dissociate between acquisition and expression of knowledge. And we trained freely moving rats and head-fixed ferrets on distinct behavioral tasks. And consistently, we found that these non-reinforced probe trials revealed strong latent knowledge, regardless of the specific task used, the sensory modality used, whether the animal restraint approach, or whether reinforcement was appetitive or aversive. And I think that a big challenge in evaluating behavioral data and constructing good models of learning is also that the learning trajectories of individual animals often appear so variable. And for many of us, certainly including myself, this has long been thought to be due to differences in how fast animals make sensory motor associations. Smart animals learn quickly, whereas animals that learn more slowly are generally thought of as less capable. Armed with this dissociation, we decided to explore the variability in learning curves. And in the presence of reinforcement, we indeed find this huge spread in how long animals take to learn the task. Some animals learn the task very well within 4,000 trials, and some take up to 8,000. On these non-reinforced probe trials, though, this variability was strongly suppressed, with all animals acquiring the correct sensory motor association in a roughly similar time frame. And this makes us think that the performance variance is not a result of differences in how fast animals make association or the underlying learning rates, but rather a contextually mediated effect. We then decided to explore what computational mechanisms might underlie this dissociation between acquisition and expression of task knowledge, and we constructed a neurally inspired reinforcement network model. And this consists of three three sensory populations, one that is responsive to the target tone, one responsive to the foil tone, and one that is tone responsive but not discriminating. Each of these send excitatory projections onto a decision neuron and also excitatory projections onto an inhibitory neuron, which provides feed-forward inhibition onto the decision neuron. The net level of activation of the decision neuron dictates the response of the network, and depending on the outcome of that response, i.e. whether the model is rewarded or not, a prediction error is sent back to these plastic synapses emerging from the sensory population. We hypothesized that learning occurs on these reinforced trials, whereas in the probe context, the model continues to operate, but no feedback is sent to the synapses. Instead, we hypothesized that the difference in behavior between these two contexts was mediated by a modulation of the readout effect, and this would allow for a difference at the level of output without changing the synaptic weights between the two contexts. So essentially, the model will operate solely based on the weight matrix in the probe context, whereas a scaling factor would be applied in the presence of reinforcement. And we searched for a biologically plausible mechanism that we could contextually modulate the readout function. And one idea was that we would have some kind of additive threshold shift. The threshold to response in the decision neuron would be increased or decreased. Another was that we would essentially scale the gain of all excitatory synapses in the network. Another that we would selectively scale the gain of excitatory projections onto the decision neuron. And a final one that we would selectively scale the gain of all inhibitory synapses in the network. And surprisingly, this inhibitory scaling mechanism was the only one that could capture our behavioral results. And essentially, we would see over the course of learning, the representations of target and foil tones slowly diverge in the presence of reinforcement. But when we look on these probe trials, where we remove the scaling mechanism, we see that these representations are further diverged. And if we look at the hit and false alarm rates of mice performing this task, on average, we see that the model captures these very well, and simultaneously, without changing the underlying weights, captures the hit and false alarm rates in the probe context as well. And across 7 individual mice, we find that this inhibitory scaling mechanisms perform better than all the other tested mechanisms, and we also found that it could capture results from freely moving rats and head-fixed ferrets and other behavioral tasks. And finally, we've started to do some preliminary gather some preliminary data on how the neural populations in the auditory cortex are changing over time, as that we can identify this faster learning trajectory than performance would suggest. So we express GCAM6F in excitatory neurons in the primary auditory cortex, select regions of the auditory cortex that are responsive to our target and foil tones, and then image the same populations of neurons over the course of learning. And consistent with previous results, we find that some neurons do increase their selectivity for either the target or the foil tone on a time scale that roughly aligns with the performance improvement in the presence of reinforcement. But we also find neurons that rapidly learn to discriminate between these two tones, suggesting that the brain, or at least the auditory cortex, is fully capable of discriminating between these two different stimuli much earlier in learning than the mice's performance would suggest. And if we look at on a network level, early in learning, we again see that we can decode, using a linear classifier, the stimulus identity very early in learning on a timescale that aligns with the behavioral improvement in the probe context. But we also see changes that are more aligned with the behavioral improvement in the presence of reinforcement. So for example, a larger and larger fraction of the population seem to be selective for either the target and foil tones as learning progresses. And the network appears to be more stable, or at least stabilized only at the very late stages of learning, such that we can use the same decoder weights from one day onto another. So in summary, what I've told you today is that reinforcement feedback, of course, a critical mediator for learning, paradoxically seems to mask underlying knowledge. Mice learn faster, and mice and other animals learn faster than their performance in the presence of reinforcement suggests. The acquisition of task knowledge is actually very stereotyped, and the performance variance is likely more a result of contextual factors that are impinging on behavioral decision-making. And the acquisition and expression of knowledge may rely on distinct computational and physiological mechanisms. And with that, I'd like to thank my mentors for this work, Kuchibhotla and Froemke, my current mentor, Vanessa Ruta, Sergeant Ostojic for help with all aspects of the model, Peter Holland for rat behavioral data, Rupesh Kumar and Boubenec for ferret behavioral data. Thank you.
Thanks very much. We're well in time. Yeah, question.
Yeah, beautiful work. I have a question about your last slide. You mentioned reinforcement feedback, and these two things are coupled. So the drop of water is both feedback that the choice is correct, but also reinforcement. Do you think that if you were to give the animals purely feedback on their performance without the motivationally salient water, that you would see the same patterns? In other words, do you think it's the reinforcement or the feedback, or both, that is masking the task knowledge?
So I guess it would be a little bit hard to decouple the two, but it's a good question. I think, the reinforcement somehow probably elicits a motivation to get more tasks or get more rewards. And I think, I don't think we know if it's that motivation that's driving the change in behavior, or if it's the presence of a single reinforcing variable. Is that your question?
I guess if you would play the animal some feedback stimulus to just tell him
whether he was correct or
he did the right thing or not.
Yeah, that's a good idea. We have not tested it. My hypothesis would be that it is the reinforcement itself, because across tasks, whether it's appetitive or aversive, whether it's shocks, it seems to be specifically the presence of reinforcement or the receiving of reinforcement that drives this change. But it's possible that we have not specifically tried delivering just a kind of answer cue.
Thanks.
And I have a complementary question. Did you try sating the animals with water and then putting them into the context with the active lick spout? I guess under the same hypothesis, is the false alarm rate high because they are basically putting a prior and trying to get the water because it's available, and they can actually distinguish between these the state of knowing the task and this other impulse, which is just to try to get the water. So did you do any kind of satiety manipulation?
No, we have not done any satiety manipulation in this task, but we've done other tasks where essentially it's a freezing task in rodents, where or in freely moving rats, so they receive a shock, a foot shock, and then we measure how much they freeze in response to one tone or another. One tone predicts the foot shock, and we see that this again dissociates, and these are not motivated by any satiety or essentially thirst. So I think that kind of gets it.
It seems like an implication of your work is that as a group we are worse and more variable when stakes are high, and that seems to have quite troubling implications. How do you interpret this in terms of the implications for everyday life?
No, I think it is true, but I think that it has a lot of implications for how we think about learning in general, and especially this decreased variability in terms of acquisition of sensory motor associations. But I'm not sure that we encounter tasks that span 5,000 trials where we can really dissociate between these. I mean, it might be a better strategy to actually not dissociate in sometimes, to not accurately perform the task because of exploration and exploitation trade-offs. But I'm not sure how we would, it's possible, but I'm not sure how we would essentially think about this in a more narrow task. We've been training humans on similar tasks as well, and our problem is that humans essentially learn tasks too quickly to do this dissociation. It seems to come into play where we have a longer time course of learning, so I'm not sure if it applies in the same.
One more?
Yeah, this is a very interesting study. I was wondering, and you might have said as much, but I was curious if there was an asymmetry between the adversive version of the task and not, given that I would expect there are sort of distinct motor mechanisms.
So there is an asymmetry. So in most appetitive versions of this task, we see the false alarm rate drop, whereas in the aversive version, we actually see the inverse essentially. So the go response is increased on probe trials instead of the suppression of the no-go response.
Hey, over
Question
here.
on the other side.
Very interesting. Have you looked at how quickly the performance improves at the start of the probe trials? Is this instantaneous as soon as the lick spout is removed?
So, I mean, it's a little bit hard because you have a limited number of trials to assess the performance, but it seems like within 1 or 2 trials of switching where the animal doesn't know that he is getting reinforcement, they stop responding to the foil tone, essentially.
Okay, let's take one last question.
Hello, this is a very interesting study. So I'm just thinking the possible interpretation of this context modulation of performance. So is it possible that this, the different contexts just, when you change the context, it just down-regulate the value of both, the both cues, but this way can increase the contrast of these two cues, so that can increase, that can boost the animal's performance.
Yeah, I mean, I think that's true. In a sense, that's what this inhibitory scaling mechanism is doing. It is scaling up it's changing the valuation of both cues, because both cues are receiving an inhibitory signal, and by modulating that, we're essentially allowing the cues to be better separated. Does that make sense?
Yes, this reminds me that's kind of similar to that, some people claim that slow wave sleep can, down-regulate the synaptic weight, for all the memories, but this way can increase the, the memory the human animals want to install. That's the most useful information. So that's kind of comparison.
Yeah, absolutely. I think that's a really interesting direction, and it would be interesting to explore. Thanks.
Okay, thanks again for a great session.